Hello, so we're going to recap today what we learnt about osmosis, just so it makes sense when you come to revise it at home later. We learned about diffusion last lesson, the movement of particles from an area of high concentration to an area of low concentration, so you end up with an even concentration in that particular system. Today we learned about osmosis being basically the same thing. It is diffusion, but it's diffusion specifically of water particles. That's worth 1 mark. Diffusion of water particles. It's worth mentioning that that's from a high concentration to a low concentration for a 2nd mark, and also that has to be across a semi-permeable membrane. So that's basically like your cells. Cell membrane around your cells, water can diffuse into your cell, and water diffuses out of your cell, and often that's happening at the same time in both directions. But overall you can get a net movement of water in or a net movement of water out. So we likened it a little bit to being in a bath or a swimming pool. Being in a swimming pool, you're in a solution which has lots of these water particles in and very little dissolved substances compared to your body. And we've actually put some potatoes into tap water. And this is our model that we drew up. We said, we think that the tap water has got a very high percentage of water particles in it. And we drew 1 salt particle or 1 dissolved particle for every 9 water particles. And we said, just hypothetically, that we'll call 90% water. So that's a very high concentration of water. When we drew our potato in here, which would be a bit like you when you're swimming, that would be a little model of you, we did 6 water particles for every 10 particles in total. The other 4 of the 10 are actually salt. And we said that that would be, say, 60%. As the model goes here. That's not quite like that in real life, but let's go with it for the model. It means that 90% water outside, either in the bath or in a swimming pool, is much higher in concentration of water compared to the water in your cells. So water is going to diffuse, in theory, from the water outside the body into the lower concentration in your body. Now the word we used was hypertonic. Hyper meaning lots of. So there's lots of water compared to the water in your cells. The percentage is higher, if you like. We also looked at the opposite side. We looked at what if you went swimming in the sea, and the sea was particularly salted, the Red Sea, the Dead Sea. There's going to be a very high percentage of salt. We also agreed in class that that would be unrealistic, but for purposes of the model, this works for us to help understand it. We drew the same proportion of water and salt in the body as we did back here, because your body's still your body. It's got the same ratio of particles. Still, on this model, 4 salt for every 10 particles, and there are 6 water particles for every 10. So we've called that 60% still. It's just the difference is outside, we did pretty much 1 salt particle for every water particle, which means out of, say, 100 particles here, 50 of them are going to be water and 50 of them are going to be salt. Again, it's not real, it's not realistic, but for the model it works. That means 50% of this solution is water. We're focusing on the water concentration, not on the other stuff that's dissolved in it. 50% water outside the body in a very salty sea would mean it's got a lower concentration of water outside the body compared to inside, which is 60% here. That means water's going to travel overall, a net movement from the high concentration in your body into the low concentration outside your body. That means your body's actually going to lose mass. It's going to lose water if you spend a lot of time in salty water. Equally, if you spend a lot of time in the bath or in a swimming pool, your cells will take on water as the water diffuses into your cells, and that's why your cells swell up and your skin starts to wrinkle. The last thing we looked at was this idea of an isotonic solution. We're going to find out which concentration from our experiment we did today, which of these concentrations of salt in the water provided us an isotonic solution. That would be an equal level of water, an equal concentration of water, in this case 60% outside of the body in the fluid and 60% in the living tissue. Overall, water will be diffusing in and it will be diffusing out of the living tissue. It'll just be doing that at exactly the same rate. So there will be no net change. On our results table, we put 5 lumps of potato into 5 different boiling tubes with 5 different concentrations of salty water. And we said that the tap water was 0 molar, therefore that was the highest concentration of water. That was our hypertonic solution. Okay, so hypertonic there. And then we've had different salt solutions that get stronger and stronger in concentration, or higher and higher in concentration of salt, meaning the concentration of water in each of these is lower and lower the more salt we have dissolved in it. Our most concentrated salt solution was 1.25 molar, and that we called our hypotonic solution. You just have to trust me on that. When we get our results, we should be able to see whether this indeed has actually happened. The prediction is, we think in tap water, we think that our chip is going to have got bigger because water will diffuse into the tissue from the high concentration outside into the lower concentration inside the chip, and that our chip may go, say, from 20 millimetres in length up to 22. Now that would be an increase of 10%. We're then going to plot this on a graph next time. At the other end of the scale, we thought that our salty water was hypotonic, so this model here, we think there will be a higher proportion of water molecules inside the potato cells compared to outside. That means this time water is moving from inside the cell to the outside. The potatoes would physically get smaller, they would be way less, there'd be less mass at the end, they would get smaller from 20 millimetres, maybe they'd go down to 18, just as an example. That would be a 10% loss. So we'll get to plot that on a graph next time. I hope that makes sense of hypotonic, hypertonic and isotonic. So we have our results of our osmosis practical, and we've just sketched those up here. From the salt concentration going from 0 up to 1.25, we found that the potatoes in the low concentrations of salt and in the tap water here have actually gone up. We worked out the percentage increase and plotted it on the graph. So let's just plot the graph here. This is quite useful because we can see on the graph that at the lower salt concentrations, our potatoes have increased in size. That suggests that water's actually gone into the potato, diffused in. Hypertonic solution outside the potato, and that causes higher concentration of water outside, diffusing into the chip. The bigger the difference is between the salt concentration, or particularly the water concentration outside the chip compared to the concentration of water inside, the bigger the change is. We end up here with our hypertonic solutions getting down to a point where, had we actually used that concentration, which was around about 0.8 as an estimate on this graph, 0.8 molar, we would have ended up with isotonic. That means that the concentration of water inside and outside the chip would have been the same. Water particles would be diffusing out of the chip and into the chip at exactly the same rate, so there's no overall net movement. Up here in the hypertonic solution, the net movement is water diffusing into the chip. That's why they got bigger. Go above the isotonic point and we see that the chips actually get smaller in length. Their percentage change is a negative number, and the bigger that difference is, the higher the concentration goes, the bigger the difference is between the water concentration in the chip and the water concentration outside of the chip, meaning water is actually being lost, and the more salty the water becomes, the more water is actually lost to the outside of the chip. That means we've got hypotonic solutions over this side of the graph. One other thing to spot, which is just more to do with graph construction, is we've got a point down here which doesn't really seem to fit this pattern of data, which is normally for this sort of experiment a curve going down in this kind of shape. So we've just highlighted that, an anomalous result, or also known as an outlier. That there, hopefully, if you look back at the earlier part of the video with our models, particle models of hypertonic, isotonic and hypotonic solution, should make a little bit of sense.